I'm delighted to have you join us for the first of two Dean's Distinguished lectures for the School of Medicine connected to our reunion weekend. And this year we have a prominent School of Medicine graduate studies classes of 1984 alumnus who received his degree in molecular biology. And with Fritz Rotman, Richard Woychik, who is with us celebrating his 40th reunion from the Case Western Reserve University School of Medicine. I'm delighted he was able to join us. Rick became the director of the National Institute of Environmental Health Sciences and the National Toxicology Program in 2020. And in those roles, he gets to oversee all of the aspects of both funding and internal research programs and advising on biomedical research in the space of environmental health sciences and toxicology. He served as a deputy director at the NIEHS since 2011, working with Sam Wilson. I happen to interact a lot with Sam Wilson because of our common interest in basic excision repair, although I will say that he usually told me I was thinking about it wrong, and he advised me quite well about a better way to approach the questions I was asking in an incredibly, as Rick understands, patient manner of offering really good advice by saying, well, I wish you'd read the literature better, but always coming away from those conversations better informed. So, and I had 3 or 4 trips down specifically to spend a day with Sam in that 2007 to 2015 period where we were actively studying basic excision repair. I'm going to share with you the mission of the NIEHS because I think it's important to understand and conduct research into how environment affects biological processes across the lifespan, and to translate our knowledge to reduce disease and promote human health, and to provide global leadership for innovative research that improves the health of people and communities. I have to tell you, Rick, it's actually remarkably close to the mission of the School of Medicine, so thank you. Exactly. So his laboratory was, in fact, the first to clone the gene in mice called agouti to give us better insight into obesity and how we managed our food intake. He also studied a gene associated with polycystic kidney disease, studied genes involved in protocadherin and hearing loss. And I think it's curious that, that background in understanding genetic disorders in mice led him to become the president and the CEO of Jackson Lab in Bar Harbor. And that laboratory, he helped become the director, or helped become an NCI-designated cancer research center. And that, I think, helped lead him into having the pathways and mechanisms and model systems to understand environmental toxicology and environmental health. I should point out that after he left here with a PhD, he went on to work with Phil Leader at Harvard Medical School, which, if I remember correctly, Tony Winshaw-Boris followed soon thereafter, and also had fellowship support from the Howard Hughes. So without further ado, as you can tell, I'm incredibly enthusiastic that Rick is here, and to recognize that he wouldn't have his current position if he hadn't graduated from our medical school.
Thank you.
We get to be careful with our federal employees, but this is a small little gift recognition. It says Richard Woychik, PhD, class of '84, Dean's Distinguished Lecturer, November 14th.
Well, thank you very much, and I will display this proudly in my office at NIEHS. Stan, thanks very much. Well, and I'm really excited to be back here in Cleveland, and I'm excited to be back at Case Western Reserve and to have an opportunity to meet with old colleagues and old friends. It's been a really exciting time. But I have to say that most of all, I'm really excited to be here to share with all of you the strategic plan that I just rolled out to our council last September. And so I'd like to take about 30 minutes today to go over some of the areas of scientific focus that we feel will really set the environmental health sciences community up to not just make incremental changes in the status quo, but we're hoping that these will be the types of things that will truly propel environmental health sciences into very exciting new directions. And most importantly, something I'm personally very passionate about is how do we get environmental health sciences integrated into the fabric of the way that we study the etiology of human disease. Because you'll see in just a few minutes, it can't be just about genes and genetics and genomics. There are environmental perturbations that can affect how our genes are working. And these environmental exposures may be interacting closely with our genetics and our genomics. So we need to change the way that we think about things. We need to create a collaborative environment. And in many ways, I think part of the reason why Francis Collins hired me for this job is that I'm pretty much following the playbook that he used at NHGRI and with genetics and genomics. So back if you were doing genetics and genomics 30 years ago, you were probably at NHGRI or you were funded by the Genome Research Institute. But through his leadership and Eric Green and others, genetics and genomics has been proliferating. It's integrated across the biomedical spectrum. So people in environmental health sciences use genetics and genomics, and it's part of the reason why I'm the deputy director and I'm the director. So I want to do the same thing with environmental health sciences. We need to get it out into across the NIH and across the biomedical spectrum because it's really important. So let me just start with just a couple of minutes here talking about NIEHS. So some of you I know about NIEHS because you get money from us, and you probably read the strategic plan. You're really smart to do that. But for those of you who do not know about NIEHS, we are the National Institute of Environmental Health Sciences. We are one of the 27 institutes and centers, and I'll talk about ICs occasionally during my presentation. So it's institutes or centers. Now, if you're coming to visit us, be very careful. We're not located in the greater Washington, D.C. area like 26 other institutes. So if you take the metro from the Red Line to Metro Center, you have about a 350-mile walk to come to us in North Carolina. So why are we in North Carolina? Well, it turns the short answer is that in the 1960 election, Governor Sanford in North Carolina delivered North Carolina, the Republicans in North Carolina, to Kennedy for the election. So when the governor had this bold idea to essentially create the Silicon Valley of North Carolina, it's called Research Triangle Park. Some of you may have heard of this. So they needed an anchor tenant. We're the anchor tenant. So I'm pleased to say that NIEHS was there. We then provided the means to attract IBM and several other companies. And again, it's transformed the economy within North Carolina. And it's actually a very exciting place to be. So there's phenomenal biomedical research that happens at the University of North Carolina, Duke, at North Carolina State, Wake Forest. So it's a very rich research environment to be doing the types of things we do. Now we have intramural research laboratories. There are about 55 different PIs who study basic mechanisms of how do human beings work at the molecular level. And we also have an applied research division, intramural division, where we actually take some of those principles from our basic research laboratories and build innovative new tests for toxicology science. Now, the bulk of our budget gets wrapped up in the form of contracts and grants, and we package it up and send it off to Case Western Reserve and other places primarily across the United States. So we're not the biggest and we're not the smallest, so we have a budget of about $1 billion. So some of the other, the NCI is a little bigger than we are, but part of my motivation is to use the $1 billion that we have in a way that we can leverage what we fund with work that happens in other ICs. So we can bring environmental health sciences into the fabric of studying the etiology of human disease. So maybe some of that $6 billion now at the NCI, whatever it is, maybe we can carve out a little bit of that and help to support work in support of environmental health sciences. So Stan very nicely presented the mission and the vision. I think it's the mission and the vision that differentiates NIEHS from other institutes and centers across the NIH. And this is really all about studying the impact of the environment. So what I can do is tell you, well, why do we want to study the environment? Actually, my physician has never asked me what I'm exposed to in the environment, never asked me where do I work, where do I live. That's a problem. So if you're physicians in the audience, ask your patients where do they live, where do they work. Do they work in a place that is likely to be exposing them to certain things that may be associated with the health conditions that they're complaining about? So it's very important. In fact, if you look at some of the publications here, Rappaport and Smith back in 2010 are estimating that 70 to 90% of disease risks are linked to differences in the environment. Now, that's not just exclusive of the environment. I mean, there are a lot of gene by environment effects. I mean, our genetic basis can be influencing how we respond to the environment. So it's not a great analogy, but often it's said that the genetics loads the gun and environment pulls the trigger. So we have to, and again, I'm trying to come up with a better analogy, so if anyone has one, talk to me afterward. But it actually points to there is a role for genetics and there's a role for the environment. So what are some of those environmental triggers that may be responsible? Just because you're genetically set up to develop cardiovascular disease or type 2 diabetes doesn't mean you're going to develop it. So could there be other modifier genes that we haven't located yet? Or are there environmental triggers that we need to be paying attention to? So the environment is really important. And in fact, if you look at just a number of different exposures, I'll just quickly go through this. Lead, arsenic, you have PFAS. You've probably heard about PFAS in Ohio. Perfluorinated alkyl substances. You know, the Teflon that we've generated hundreds of millions of tons of this stuff. This is persistent in the environment. It's never going to go away. It's going to be on the face of the earth. And there are health effects associated with PFAS. You can see this immunotoxicity. In fact, some of our grantees have demonstrated that the PFAS exposure can actually impact the response to vaccines. So there's just a lot of things that are going on. Flame retardants, we're actually, unfortunately, breathing them in from the fabric that's on the seats. Pesticides. I mean, there are pesticides. I'll talk about some of the work in my own lab in just a couple of minutes. Rotenone, pesticide targets complex 1 in the mitochondria. Very, very strongly associated with the development of Parkinson's disease. And some of our grantees are actually convinced that there are pesticide exposures that are the environmental triggers for Parkinson's disease. And of course, PM 2.5, you know, the particulate matter that is one of the most important active ingredients of air pollution. I mean, you look at this, respiratory outcomes, heart disease, autism, diabetes. So these environmental exposures are important. If you look just at air pollution, a whole bunch of different things, respiratory, cardiovascular, neurological, immune, metabolic, and neonatal. So we can't ignore the environment. And in fact, one of the things that's happening, in fact, the new director, relatively new director of the NCI, we were at the IC directors meeting a couple of weeks ago, and she leaned over and said, Rick, you know, can you help us understand why there's such a remarkable increase in the incidence of early stage cancers? So not just colorectal cancer, but a variety of other cancers. And I said, well, you know, it's probably not genetics. We don't evolve that quickly. So it's the environment. So she indicated yes, so she's going to come and we're going to talk more about this. So what are some of the environmental triggers that may be responsible for this very significant increase in early onset cancer? Same is true for autism. Same is true for a number of different diseases. So what are those things in the environment that are clearly having adverse health effects? And we need to take a look at this. So studying the environment is complicated. So here it's looking at all of the different things, agricultural chemicals and pesticides. So there are hundreds of these things. There are synthetic materials. PFAS is probably one of the most notable examples, but PFAS is an umbrella term. There are 13,000 different molecular entities that all fall under the PFAS umbrella. So we need to better understand, so those 8 carbon molecules that were originally generated to create the first generation of Teflon, they're really dangerous. But are the 3 carbon molecules or some of the other species that are still currently being made by DuPont and other companies, are they as dangerous as some of the original 8 carbons? So disasters and wildfires. I mean, I think we're all beginning to appreciate breathing, especially if you're living in New Jersey at the moment. You probably have a great appreciation for the potential impact on your human health of some of the wildfires that are happening. In fact, you know, one of the things we were talking about today,
wildfire smoke isn't necessarily just burning trees and bushes. If you're now burning communities and you're burning down auto repair shops, you're talking about urban wildfires are just different than wildfires had happened in the forests in Canada. So it's very complicated. So we need to be teasing apart. What are the impacts of all these things? And plus, we need to be looking at diet, lifestyle, exercise, stress. These are all critically important components. In fact, are some of the, say, good exercise, not just lack of exercise, but good diet. Could good diet, omega-3 fatty acids, polyphenols in blackberries, can they mitigate some of the negative effects associated with some of the other exposures? So these are things we need to be looking at. And of course, part of the reason why I'm interested in this is that different individuals with different genetic backgrounds respond to a chemical stimulus or an environmental stimulus in different ways. And so in the medical community, we talk about precision medicine, where we're now sequencing the entire genome and designing therapies based on an individual's genomic sequence. Well, in the environmental health sciences community, we need to get away from this whole business. We do everything based on statistical averages across populations. So let's bring that same sophistication to the way that we do environmental health sciences. So we look at things in the environment, we actually look at genetic background, and we look at individual biological variability, and then we create strategies to integrate both of these things together. So take a look at the strategic plan. I have limited time today, so I'm not going to go through all these things in great detail, but I'm going to focus on, there are 6 different areas of scientific focus that have emerged from our strategic plan. Again, these are the things that we're hearing from, well, some of them I'm personally passionate about, and some of these things are coming in from the world community, that we need to be dedicating time and effort and resources to help to make these things happen. So let me go through those now. I'll take you through each of these 6 areas of scientific focus. So they're listed here, but let me go through them one by one. So let's start off with exposomics. Okay, so exposomics. What is the exposome? So some of you may have heard about this, some of you probably haven't, but it was back in 2005 that Chris Wild, an environmental health scientist, was watching the release of the genome sequence. So the genome sequence was published in, 2002? And the genome sequence was really all about, if we really want to, study genetics, we really have to get beyond one gene at a time. So if we have the genome framework, we can study the entire genome, looking at all the genes. And that is, and we've discovered that's been transformative in the way that we do genetics. So he actually made the comment that we need to do the same thing with the environment. So we're exposed to all these different things that I showed you in a couple of slides ago, and all of them are affecting our biology, not just one thing. So if we're studying just PM 2.5 and air pollution, well, we're not really factoring in the contributions to our biology from PFAS, from flame retardants. So we need to develop a different strategy. We need to be thinking about the environment in its totality. In fact, he defined it as it's the totality of exposures over the life course. And it has to be over the life course because, in fact, I'll show you some data from my own lab, but we know that there are things that, my mother and your mothers are exposed to during pregnancy that can actually affect how your genes are expressed, not just during development, but actually affect how your genes are expressed much later in life. In fact, some of these things are heritable across the generations. So we need to come up with a strategy where we look at these things, the totality of exposures over the life course. Now, the totality of exposures, you have physical chemical exposures. We have pesticides, POPs. We have plastics, plasticizers, the micro and nanoplastics that are in the news. We need to figure out how we can actually assess the biological effect of all these different things, not one at a time. But it can't be just physical and chemical exposures. The exposome also includes social, lifestyle, and ecosystems. Diet, exercise, psychosocial stress. Psychosocial stress, as I think we all know, can be really bad. And lack of psychosocial stress is actually potentially a really good thing. So that's the exposome. It contains all these different elements here. So it's a big, bold vision that if we truly want to understand the environment, it's the totality of exposures over the life course. Now the challenge, of course, is it actually has good mouthfeel to it. But how do we do studies using an exposome framework? What data do we collect? How do we share this data across the global community in a way that experiments And studies that are being done in one country can be seamlessly shared with studies in another country. So we need to operationalize the exposome. So just like with the Genome Project, it started with a big, bold vision. And I have to say, back in the early days, I don't think anyone knew how we were ultimately going to sequence the genome and what technologies we would ultimately have available to us so individual genomes can be sequenced and put as part of your medical record. But we have the big, bold vision here that that's what the exposome is. And we may have to build new technologies to do this, just like with the Genome Project. I remember some of the early, I don't know if you remember the cDNA clones on microscope slides. I mean, that was a big innovation of, that was the way that we can look at multiple different genes at once. Well, that's primitive compared to what we can do today with RNA-seq. So we may need new technology. But on the other hand, just like with the Genome Project, we can do what we call the pragmatic exposome. There are things we can do right now. In fact, we have some remarkable tools, high-resolution mass spectrometry. We can take a small sample of blood, and we can analyze that and very accurately estimate all the different chemical entities that reside in your blood. And if we do this over a time course, we have a pretty good sense of what the turnover is. So let's start with some of these technologies. And so what I bring to this is, again, the model from the genomics community. I mean, we started off with a bold vision in the genetics and genomics community. We had to develop new technologies, where we kept the focus on that end game. What is it that we ultimately wanted to achieve? Now, it was a whole genome sequence. So we actually then developed that strategy. We need to do the same thing with the exposome. So how are we going to make this happen? So there's a lot of work that needs to be done. And so I have this simple mantra that things get done when it's someone's job to get it done. So what we've recently done is we've funded a center. It's a collaboration between Columbia, Harvard, and the University of Southern California. And the idea is to essentially build a community of practice for cooperation and collaboration, to define how to operationalize the exposome. In fact, I spent a couple of weeks in Europe a couple of weeks ago, and there's a lot of discussions that are going on right now about how do we operationalize this? How do we collect data? How is an exposome, a study using the exposome framework, different than a study where you're studying one thing at a time? And should everything be done with an exposome framework? Probably not. There's still a role for studying one thing at a time. So we have the center. It's called the Nexus Center. And a lot of what you're going to be hearing me talk about is collaboration, building new partnerships where we bring people with complementary skill sets together, working collaboratively to make bigger things happen. So you can actually Google the Nexus Center at Columbia and learn more about the details. So let me move on to the next area, which is precision environmental health. So what is precision environmental health? You probably heard of precision medicine. So precision medicine is about sequencing everyone's genome, looking for sequence variations that we inherit from our parents, maybe spotting a spontaneous mutation here or there, and then correlating your genome sequence with health outcomes. Well, so precision environmental health is all about trying to account for individual variability. What may be exquisitely dangerous for me may not apply to you. And furthermore, it's better understanding the etiology of say, type 2 diabetes. Was it caused by genes, or were there interactions with the environment? So precision environmental health builds on the framework of precision medicine. So you start with whole genome sequence and a lot of the genomics tools, but you overlay it with environment using an exposomics framework. So it's really, again, about the bottom line is really about trying to develop a framework to study those gene by environment effects. As I showed you on one of the previous slides, it's probably the vast majority of disease and health-related issues in humans are gene by environment effects. So this is the framework that we need to use for that purpose. So one of our grantees actually talked about precision environmental health as being G by E by D. So it's genomics by environment. By D is data, because as you can imagine, when you're taking a whole genome sequence, looking at the totality of exposures over the life course, there's a lot of data, okay? And we need to be thinking about how do we manage this data in a way that we can kind of synthesize this and actually develop strategies to better understand the impact of the environment and genetics. Now, the genetics part of this is complicated too, because there's no single gene that's responsible for, well, actually maybe in a couple of cases, but the vast majority of cases, the genetic susceptibility to either adverse health effects or no health effects. These are complex traits. So there are potentially hundreds, if not thousands, of different gene variants that we each have that gives us this differential susceptibility to an environmental exposure. So the best way to do this is to actually join forces with the genetics and genomics community. So many years ago, Francis Collins and Eric Lander called a meeting at the NIH to develop something called the International Common Disease Alliance. So that was the recognition that if we really want to tease out the hundreds, if not thousands of different sequence variations associated with complex traits of common disease, type 2 diabetes, obesity, a variety of different things, let's be smart about this. So instead of everyone working in a vacuum and in a silo, let's start developing standardized ways that we collect our data. So the work that's done in the United States can be seamlessly integrated with the work that's being done in Europe and in Iceland and in other parts of the world. So that was the ICDA. But at the meeting, I actually raised my hand and said, well, Francis, what about the environment? I think we maybe better factor the environment into the equation here. So that's what we've been doing. In fact, I've been working together with the CEO of the All of Us program. So I'll get into that in just a second, but we've been working together with people in the ICDA to integrate environmental exposures into the data that we collect to assess these complex traits. So the other thing I just want to point out here is that the E part is not just environmental exposures, but many of the environmental exposures actually change epigenetic signatures on your genome. And what that means is you're not mutating the DNA or changing the nucleotide sequence, but you're actually chemically modifying the nucleotides. And you're doing it in a way that you can change the way that genes are expressed. So you can either change the way genes are expressed by mutating the DNA, or you can change the way genes are expressed by these chemical modifications, the methylations of C, for example. So it's epigenetics. There are also modifications of histones. So we need to factor epigenetics into the equation. So why is that important? So this is the slide actually from my own lab. This is, Stan referred to that agouti gene. That agouti gene is a coat color gene. But as you can see here, if you mutate that agouti gene in specific ways, you can actually make mice fully yellow and make them fat. So in fact, the mouse here on the left is an 85-gram mouse. It's a litter mate with the mouse on the right. Okay, they're the same age, and one of them is 85 grams compared to a 25-gram litter mate, and the difference in size is adipose tissue. So how does that happen? So you might look at this and say, well, these different mice, they look different. Some of them are mottled, with yellow fur and a agouti background. So they must be genetically different. Okay, they're not. All these mice are genetically the same, at least in the nuclear genome. They are genetically the same, and they all carry the mutation that confers the yellow coat color and the obesity phenotype. So how is that happening? Well, it turns out that the mice that are phenotypically normal, the body has epigenetically silenced the mutant form of the gene. So the mutant form of the gene is not being expressed. So one of the questions we asked is, so this, I guess I call this epigenetics in action. You want to know why epigenetics is important? Take a look at this. It's not just genetics. Genetics plays a role. If they didn't have the mutation, they wouldn't be yellow and fat, but it's the environment that can be influencing this. So one of the things we did is, in fact, this is part of the reason why I'm very interested in environmental health sciences. So the distribution of phenotypes here is influenced by what you feed mothers. In this case, what you feed the females during pregnancy. So you can shift the phenotypes. You can create more mice that are yellow and obese if you feed them or you expose them to certain chemicals. And in fact, that's what we did. A few years ago, we published this in Cell Reports. So we actually treated them with a mitochondrial toxicant, a pesticide. And you can see here, you increase the number of animals that are actually fully yellow and obese. So we asked the question, is agouti the only gene in the genome that is responding to these environmental perturbations? And the answer is no. So we did something called whole genome bisulfite sequencing. And this is a lot of detail here I'm not going to go into. But you can see that this is a heat map. And there are different blocks of genes at different times during development. Day 22 is birth. 6 months, 12 months, and 18 months. So there are blocks of genes that are turned on and off over the course of development and aging. And treatment of 10 parts per million with this pesticide actually interferes with the ability of those genes to be expressed. Not just at day 22, which is when we stop the rotenone treatment, but there are things that are happening at 18 months of age, which is a very old mouse, as a consequence of the exposure to this pesticide during development. So you got to pay attention to the epigenetic part of this too. And in the end, this is going to be a huge challenge to integrate lots of different data together. You know, the microbiome plays a very important role, the proteome, the metabolome, transcriptome, the epigenome and the genome. The exposome contains all of these things. So we've got, you know, huge challenges in terms of data management. Data collection in a way that it can be systematically compared, and then data management. So I also want to talk about another area of focus, which is in climate change and health. So soon after the last election, President Biden made it very clear that, that climate change is a high priority for his administration. We won't talk about where we are today, but it's, so he issued Executive Order 14008. And so he actually brought $40 million to NIEHS. We had actually done quite a bit of work studying the health effects of climate change. Not the, you know, some of the politically sensitive issues in climate change, but what are the health effects of climate change? So we recognize, I recognize right at the outset, this is much bigger than what we can do on our own. So let's put forward a different collaboration model. So there are people at the National Institute of Mental Health. There are mental health consequences of climate change. I know that actually the former director of NIMH, you know, his kids, his teenage kids, are really upset about what we're doing to our planet. So we need to be studying all the different aspects of this. So what we did was put together a collaborative effort involving multiple different institutes and centers. You can see the names here. So NICHD, Child Health and Human Development, NIAMS, NHLBI. So we're doing this because there are lots of direct and indirect effects of climate change. There's heat-related illness. So it's pretty sobering how little we know about the extended heat impacts on humans, especially if you're pregnant, if you're a child. Say that you're on illicit drugs, working in some of the part of the migrant farm communities in California, the Central Valley of California. How does heat, extreme heat from climate change actually affecting your physiology? And then there are indirect effects from climate change. So what happens if you're if you're actually dependent, you're in a clinical trial, and the hurricane comes in and completely wipes out the medical infrastructure, as it has in certain communities in the west coast of Florida. So say that you're on dialysis. So you're dependent every 2 days or every other day. You got to go in and clean your system. So if the dialysis center is wiped out, what are you going to do? So that's clearly a health effect. So what we've done is actually, you can take a look at the website here. We've developed a strategy, an initiative around climate change and health. You can take a look at the website, climateandhealth.nih.gov. And there are some core components to this, but lots of different areas of science that need to be addressed. And we're hoping to do this in this collaborative mode. So some of the money comes from the $40 million that NIEHS got, but we're hoping that most of the money will be coming from individual institutes and centers that are working collaboratively with us on this project. Now, something you may be interested in is climate change and health, or the health effects of climate change. This is a relatively new thing for many of the institutes and centers of the NIH. So what we did was we developed something called the Climate Change and Health Scholars Program. So this is a little bit of money that we put forward. So if you're interested, you're passionate about the issues of health effects of climate change, we've developed a mechanism. It's a competitive mechanism you can apply, but you can come in to the NIH either virtually or in person, and then bring your knowledge and bring your passion and help us develop the types of research initiatives that will help us develop traction on studying the health effects of climate change. So mechanistic biology and toxicology, I just have a few minutes left, so I'm just going to go through this very quickly. So this whole area of scientific focus is getting away from the notion that if you're a toxicologist, you just buy up a bunch of outbred rats and shoot them up with different chemicals at different doses, and you count lumps and bumps and deaths over the 2 years. And that's a standard, what we call the 2-year bioassay. Well, the new vision here is now developing strategies where we can actually understand if a chemical is having an adverse health effect, how is it having an adverse health effect? What are the biochemical pathways? What are the genes that are being affected? Many of these chemicals actually act as hormones. And how are the, PFAS is an endocrine disrupting chemical. How is that hormonal effect actually influencing development and the biology of the individual? So it's really all about getting at the mechanistic underpinnings. And a big part of this, I know I spoke with several of you, there's a new initiative. It's called the Common Fund Initiative. If you don't know about Common Fund initiatives, Google it, because there is some serious money connected with Common Fund initiatives. So in this case, it's $35 to $40 million per year, potentially for 10 years. And this, so much of this is about taking iPS cells, induced pluripotent stem cells, and differentiating them into 3D microphysiological systems. So instead of doing toxicology testing in outbred rats, you're actually now doing it in a very well-defined genetic background in iPS cells that are differentiated into, say, beating cardiomyocytes or other 3D microphysiological systems. So we really hope that that will help us get to give us those insights into mechanisms of toxicology. So data science and computational biology, I don't think there's much I need to say. Hopefully it's abundantly clear to you. Everything that I've told you about goes beyond carefully taking notes in your laboratory notebook and sharing on a publication. It's about managing huge data repositories. This is expensive, so we've actually created the Office of Environmental Science Cyber Infrastructure, OESC. We got an acronym. And this is about developing plans. How do we manage these data? In fact, part of my evening and weekend jobs with our NIH director is to work across the NIH and to develop strategies. How do we manage our data across 27 different ICs? Right now, it's pretty much everyone's on their own, and we're developing databases that don't talk with each other. The data is highly heterogeneous. We need to do better. So and the final thing here is environmental health disparities and environmental justice. So NIEHS has been a leader in studying the communities, and you have many of them here in Cleveland. These are the communities that are located next to toxic waste dumps. These are the communities that are located next to the freeways. They're breathing in high levels of air pollution. So we need to study these communities. We need to generate the data so that the local, state, and health agencies that actually can provide funds to clean up these communities actually have the data in hand to make intelligent decisions on what we can do to fix the problems. So there are a number of initiatives across the NIH. I don't have time to go into details on this, but it's an area of focus for us. So and I spoke to many of you this afternoon, so I'm very encouraged that many of you know about and appreciate and are interested in getting out there and talking about the things we can do in environmental justice communities. So the last thing I'll talk about is one environmental justice community just down the road here in East Palestine, Ohio. So I think most of you know that it was actually back in February of 2023, there was a train wreck. I mean, a real train wreck. And there were 38 different derailed cars, many of them spilling vinyl chloride, benzene, isobutylene, and a number of different chemicals into the environment. So that was back in February 3rd of 2023. NIEHS, together with several other institutes and centers of the NIH, actually put together a NASEM workshop, the National Academy of Sciences workshop. And the idea is, what can we do in these cases? You have all these complicated chemicals, and you have now environmental justice communities that don't have resources. I mean, they don't have the medical facilities to actually treat some of the people who have been exposed to these chemicals. So what can we do? So we developed a plan. Part of what we've done is we provided some funding in the form of some R21 grants. And then, so we're continuing to work on how do we develop an all-of-government approach? So it's not just NIEHS, it's not just all of the NIH, but how do we partner with the EPA? How do we partner with ATSDR? How do we partner with other organizations to put together a comprehensive plan to address some of the complicated issues in these environmental justice communities like East Palestine? So I'm very pleased. Where's Fred? So congratulations to Fred for getting one of the R21 grants. And actually, I don't have time to go into all the details. I'm sure he can tell you all of the details, but I was very encouraged that there are lots of really good thinking about how we can bring multidisciplinary approaches to address the challenges in these environmental justice communities, because it's not simple. And it's certainly not about just studying one thing at a time. So again, I'll end here by saying we are continuing to work So it's not just NIEHS, but we have now reached out to multiple different federal agencies, including ATSDR, EPA, state and local officials. We need a comprehensive plan of what are we going to be doing to get in there and to correct and to generate the data and help to correct the problems in East Palestine. So thank you very much, and I am happy to address any and all questions that you may have. Sorry for running a couple minutes over. Yes, sir. So we're being recorded, so do we have a microphone? I want to make sure that those of you who...
Fantastic talk. Thank you very much. So first, I'm very happy to nominate you as the next science advisor to the president for the coming administration.
Okay.
So, and I'm not kidding. So, beyond the science, what mechanisms are there for an institute like yours to preach to the skeptical?
Yep. Well, okay, so many different ways of doing that. And so one of the things I do, I meet with Congress on a regular basis. Okay, so most recently I met on the issue of micro nanoplastics. They actually called me in. What are we doing on micro nanoplastics? And that's an area we need to do more. But I meet with members of Congress. I meet with, groups of members of Congress. In fact, I was telling some of you about an event at the Library of Congress. So we had several different senators, representatives, Republicans and Democrats, coming in and having an opportunity to discuss what we do. Dr. Bertinelli, she is brilliant when it comes to interacting with members of Congress. So she laid out some of the vision that she has. And a big part of her vision is getting the NIH to be working more collaboratively together, which fits very nicely with my vision for NIEHS. So we're getting out there. We're talking with members of Congress, and so I've been very actively involved in traveling, meeting with people in different agencies. When we went to, I spent a week in the Central Valley of California, really firsthand getting a good sense of what are these environmental justice communities are they experiencing, and it's actually it tears your heart out to see what some of these people are experiencing. They know. Some of these migrant communities, they know they're going to develop cancer at some point because they're being exposed to decades of residues of pesticides in their drinking water. And their kids are playing in the front lawn, and the planes are flying overhead and spraying the pesticides. And it exacerbates their asthma, and there's no one in the community to write a prescription for an inhaler. So it's getting out there, working with these communities. But before I went, I thought, okay, NIEHS in Congress has made it clear we do research. Okay, I can't go in and clean up the communities. So I said, okay, I'm fine, I'm going to go, but I want the local, state, and federal officials from the EPA, from the toxic chemical offices, and others there with me. And then before we leave at the end of the day, we're going to come up with a collective plan on what we can be doing together. So that's the all of government approach. So that's just I'll start with those things. But clearly the communication. There are some sectors of Congress, they're coming in and saying, well, we need to reorganize the NIH, we need to create 15 different institutes from the 27 that we have, and we need to do this because they don't work together. They're working in silos. So when I go and talk about the climate change and health program, I still remember when Senator Collins' staff, after my presentation, she said, my God, you really are working together. And so when I go and meet with them, I don't go myself. I bring Eliseo Perestabli at NIMHD with me. I bring Josh Gordon, the director of NIMH. I bring Diana Bianchi. We're all there together talking about what we're doing collaboratively together. So I think this is a message that's well received. We'll have to figure out, where are we? This is the week after the election. There are lots of things that are happening. But Dr. Bertinelli and Dr. Tabak, the deputy director of the NIH, reminded us that we've been through transitions of presidents in the past. We've been through a transition with Mr. Trump in the past. And so their advice to us, and the advice I'm giving our staff, is let's focus on the science. We have a strategic plan. There are important things we need to be doing. And we're gonna continue to focus on what needs to get done to improve public health. Does that answer your question?
Yeah, thank you.
Okay, other questions? Tony, are you raising your hand?
Well, Rick, I applaud you on setting out a great vision for how to figure out what's going on in the exposome and how the environment is
See,
important.
now you know what the exposome is, right?
I do.
Yeah.
Well, I've heard you talk about it before. The issue, I guess, is that it seems almost infinite. At least in the genome, you had
Right.
a certain number of chromosomes, 3 billion base pairs, and that's pretty much what you needed
Right.
to know to make the genome. But the exposome
Right.
could be a small amount, could be things you can measure, but it may be unmeasurable things. So I think you're right about saying you need to develop new strategies or new technologies, but what might some of those things be now to think about?
Well, it's a great question. So that's why where we are right now is we've got a bold vision, okay? But it can't be everything about everything about everything all the time. So that's why the operationalizing the exposome is, well, we have to define as a community what data do we collect, okay? And how do we share this data? And do we want to focus on, say, high-resolution mass spec? Okay, so you can go in now with these incredibly powerful mass spectrometers. You know, with a very small volume of blood, we can tell you all about metabolites that reside within your blood, as well as the exogenous chemicals and exogenous metabolites from those chemicals. And we could do that over the course of time. So that would be one, I think that's emerging as one very important data set. But also we need to become much more sophisticated about the social determinants. So what about psychosocial stress? I mean, we have, you know, there's data emerging that there are epigenetic changes in your genome associated with psychosocial stress. And not everyone in a neighborhood responds to, you know, an environmental stress in the same way. So can we come up with different strategies to actually monitor psychosocial stress? Can we come up with strategies to look at quality sleep? Okay, so that's all over the place, and that's very, very much individualistic. So, and we also, I talked with Dr. Alagrama this morning about noise. You know, that noise is a very important part of the exposome, and it's can not only bad noise and loud noise can not only destroy your hearing, but there are actually connections with, in fact, NHLBI funds a number of experiments to study the impact of noise as it relates to adverse cardiovascular outcomes. So that's part of the strategy, is that we funded the center. Their job is to come up with an operational strategy. And we have to define, what do we collect? Now, if we collect, we decide we do high-resolution mass spec, that's still relatively expensive and it's not high throughput. It's kind of similar to the maximum Gilbert sequencing and things we used to do. But I'm learning from people who do instrumentation development, there are these you know, totally new, innovative they're called quantum biosensors. They're not quantum dots, but they're different sensors. We can actually then put them into cells, in different organelles in the cells, and they report back you know, different things. So, and you can do this with a whole multitude of different things. So that's where we have to be thinking creatively on this. So that's where, you know, the center is about getting engineers, people who think about building machines, but present the engineers with the challenge. This is what we want to do. So, and then present them with the challenge of how do we do this with higher throughput and greater cost efficiency. And then also at the same time be thinking about data management. So what's the data architecture? So it's, you know, I use, as a genetics and genomics person, I use, you know, transcriptome as an example. So, you know, initially we had the microarrays and these differential expression. It's just a heterogeneous collection of stuff involving the transcriptome. But can we come up with a database that basically counts molecules? Okay, so that's what RNA-seq does. It counts molecules. So we can currently use the technologies we have for doing RNA-seq, but as engineers innovate on counting molecules, there may be totally new ways of counting molecules. The Oxford nanopore is a totally different way of looking at molecules within a cell. But if you can develop your database with a certain architecture, as technologies evolve, they collect the data in a way that it can be seamlessly integrated into your data infrastructure. So those are some of the things that this center is being charged with. It's a 5-year grant, but we're asking them to get out of Columbia, Harvard, and USC. Get out, talk with people at Case Western Reserve and other places. Gather information. What are your thoughts about this? And then come up with a plan that we all embrace so that we can be collecting data that's more homogeneous and that we can truly be integrating across international boundaries. Does that answer your question?
It certainly does.
It's a tough question. Sir, Kumar.
Nice talk, Rick. Just follow up on what Tony was asking. I was also amazed at the complexity that we're going to face in the outcomes, like the measures that we are looking at. I was wondering if twin studies would help in this case, at least to tease apart, like we have
Sure.
twins living apart, like in different places, so you said a whole bunch of them, and you look at factors that can be teased apart, so reduce the complexity just a little bit and compare with the general population data you're collecting.
And that would be a beautiful way of teasing out genetic from non-genetic effects. But also, just like with inbred strains of mice, that's essentially an unlimited supply of identical twins, all of the C57 Black 6J are not phenotypically the same. I mean, some of them actually have a predisposition for diet-induced obesity, and some of them don't. So that's why I think there's an opportunity. And actually, let me expand on both your question and Tony's question. I mean, the other thing I'm challenging the community with is, traditionally we think about, well, we need to develop these high throughput things to detect what's in the environment, the different chemicals that are in the environment. Well, maybe we ought to completely rethink the way we think about the exposome. So why not develop a strategy where we actually look at the biological outcomes of these complex exposures? So could the exposome actually be an epigenetic map of your genome? So you look at the sequence variations, this would be methylation changes at Cs or maybe other nucleotide changes, and that you then correlate the epigenetic signatures with complex environmental exposures. So that's, when the rubber meets the road, environmental exposures, it has a health effect when it changes how your genes are being expressed. So it's that would be the extreme case where you're completely rethinking how you go about doing this. And that puts some parameters on these. You start with the genome, sequence variations of individual genomes, but how do different environmental exposures actually influence the epigenome? And there may be adduct changes. So there are certain chemical exposures that cause adducts in DNA. So can we do the adductome? That's another term that's emerging. But in other words, thinking of the exposome not by characterizing all the stuff in the external environment, but thinking about characterizing the biological responses in individuals, that are associated with the external exposures. So, and that's why we're paying these really smart people. So Gary Miller is the PI at Columbia. Gary is actually the editor-in-chief of a new journal called Exposome. So this is a big deal. And so Dr. Patel at Harvard has done a lot of he's a bioinformatician, a computational biologist, thinks a lot about how do we manage data. And the environmental health sciences data, Tony, as you pointed out, it's complicated. So how do we manage the complication and come up with a way that we can be collecting data that we can seamlessly share? Stan, question?
So I actually find this part of your conversation quite fascinating, and as I think I mentioned to you, we had a National Academy of Medicine workshop on understanding the biologic effectors of the social determinants of health. Mm-hmm. So the emphasis you've described so far is describing the markers, the exposures, the links with some biologic factors. How do we intervene, and how do we invoke measures to know whether those interventions are successful? What sort of measures would you imagine from the data sets that come out?
Well, you start with data, and then you develop, potentially develop some metrics around what is it we want to be doing. Getting people to pay attention to the data and to alter their lifestyles in a way that can truly impact their health is, can be a challenge. But on the other hand, I guess, from my point of view, it's, I know that I, have a number of diabetes, type 2 diabetes related genes in my genome. And that's really motivational for me to pay attention to keeping my weight down and paying attention to my diet and exercising and doing those sorts of things. I don't know if you read one of Francis Collins's books, but he talked about the same thing. He did his genome analysis, and it's highly motivational. So, so it's really connecting then, how do we implement change, based on data. But it's not just, right now it's, people smoke and they live to 110. But if we can now individualize this so that, so someone who smokes, you may not be the lucky person who actually has a genomic architecture that allows you to smoke and live to 110. So is this getting at your question, or are you asking a different question?
It's that we're going to be stuck, if you will, with correlates. And I think it's not easy, but coming up with an intervention that reduces an exposure and then asking, okay, how did that affect a marker of health, and was a prediction, if you will, that if we reduce A will affect B happen? It's not so easy as you described, but designing those might be decades-long studies, or they might be short interventions.
Well, I guess the way I look at this is generating data. So when you're thinking about the exposome framework, it shouldn't be just about the collection of things that are likely to be bad for you. So, how does someone who now gets out on their bicycle on a regular basis, which is something you probably know something about, how does that actually potentially mitigate some of the negative effects associated with chemical exposures? We have some grantees at the University of Kentucky that have data, very compelling data, indicating that the polyphenols in blackberries, it's probably true of the blueberries in Maine too, there's no data on that, but that can actually mitigate some of the negative effects associated with the PCB exposure and predisposition to type 2 diabetes. So if you have now the data to connect these things, so it's not just associations, but you're actually now getting into causal mechanisms, I think that's where we want to be. Yes, question.
Yes, so I was curious to hear your thoughts on how people respond to things that they may not have control over. So for example, if I work in that field that you're talking about in California, I may not have a choice but to be exposed. I mean, I'm certain that no one wanted to move there to have their child sprayed over every day with chemicals, right? So how do we, when I think about your data architecture concept, it leads to certain kinds of solutions more likely to be found. So very individualistic or maybe medical-based solutions versus public health or policy-based solutions. And the things that would make a farmer in that community decide to spray differently might have nothing to do with his genes or her genes. So I'm just curious, how do you balance out a data architecture that allows for multiple kinds of interventions, some that might be more policy-oriented versus individual level oriented?
Okay, I'm trying to dissect the different parts of your question. So let me see here. So one of the things is, so say that you have these migrant farm workers that I visited with, who are convinced they're going to die of cancer because they have their well water is contaminated with chemicals. So one of the things, when you and I talked about this, we need community engagement. We need to have people who are affected by these exposures. We need to motivate them to actually work together with the academics and develop strategies where we can take that forward to local, state, and federal authorities who can actually make policy changes. So it's, and then hopefully bring some of these things to the farmers who are overseeing these activities. So I mean, the other thing too is there's this one person drives the tractor that sprays the fields. And I asked her, well, how do you protect yourself to make sure that you're not splashing yourself with the chemicals in the tanks behind you, and what kind of mask do you wear? I said, oh, mask? I didn't know about that. So I think as part of the strategy, you develop the data. And that, if a farmer truly is concerned about the health of their workforce, then they should, teach the person who's driving the tractor how to protect themselves by wearing a mask. Then there's the whole issue, I think you were also bringing up the issue about individualized versus population. That's kind of another issue too. So if we go in and we determine that only some people are potentially sensitive to this particular exposure, does that mean the other people, you can go ahead and spray them and it shouldn't make any difference? Well, I think the important thing about precision environmental health is really getting at the etiology of the disease. So if someone develops type 2 diabetes and it's not only their genes, but also the environmental perturbations, the environmental exposures, I mean, the way you treat that and the way you diagnose it may be different. So it's also about identifying the individuals who are exquisitely sensitive. And we know this in the environmental health sciences community. There are some people who are just exquisitely sensitive to, say, colognes and different fragrances. So can we identify those individuals and then provide counseling on what you should be doing? But it's a complicated issue, and we talked about this. There are many different variables that go into this. It's like those migrant farm workers. Well, if you know you're going to develop cancer, why are you going out? And the answer is, I got to eat. So multi-tiered solutions, and part of this we ought to be too, is how do we deal? Well, I've got to eat, and I need medical care. So how can we get in there to make sure that people who are in these communities do have the available food, healthy food, and furthermore that they have medical care. So that it was just tears your heart out that there is no medical care for these people. So it's multifaceted, and that's why we need to be doing. It's not just NIEHS, but there may be other parts of local, state, and federal government that need to be working with us to address the multifaceted problems that we're dealing with. Okay, Kumar, another question.
Yeah, I was wondering is it possible to look at the epigenetic changes in blood cells of an individual? And let's say if somebody wants to test some sort of therapy or whatever, and to go back at the individual after set time, after the therapy, to see if that epigenetics,
Right.
the levels have gone down.
Yep.
I'm just looking at, sort of following up on what Stan was asking. I was just thinking whether something like that, I don't know it'll explain everything, but perhaps a way to look at least bringing something down. And I don't know if it's how reversible those are, but that's just
some of them are, and some of them are not. And that's where, again, another approach to doing the exposome is actually going in and systematically characterizing what are the epigenetic changes associated with complex environmental exposures. And then asking the question, if you implement, say, a change in lifestyle, someone is exercising more, and once they get the diagnosis and you have the data, can you reverse some of those epigenetic changes that are clearly adversely affecting how your genes are expressed? So you can follow that, and that could be data. So you can then follow how you're doing this. So the answer to your question is yes. In fact, we have funded a couple of different centers to actually use peripheral blood lymphocytes and to ask the question, which of the epigenetic changes in peripheral blood lymphocytes reflect epigenetic changes that happen in other parts of the organism, in this case would be using mice. So we can't go in and ask someone, well, you mind if we dig in and get a part of your brain? We want to check to see if your epigenetic status is improving. So peripheral blood lymphocytes, it turns out that there are several different regions that are very robust and report back on epigenetic changes in the brain and other parts of the organism. But we also talked at one of the meetings I had about estimating biological age using epigenetic markers. So some of our grantees so there are regions across the genome that over the course of aging actually become methylated or demethylated. And so people have developed these set of epigenetic markers to estimate your biological age versus your chronological age. And so what they found is that if you're exposed to, say, those Canadian wildfires, you can actually advance your biological age And then there are consequences of that downstream. But here's a case where you actually, if you go out, you remove someone from the wildfire smoke, you can actually reverse those that advanced biological age. So a lot of different things that are happening here. But it's just thinking about how do we bring more sophistication to the way that we measure the environment. So we also talked with, I think it was with Fred and his colleagues about, the biological age is a tricky thing. It's very ambiguous. There are lots of different variables. It could be telomere length. It could be changes in the mitochondria. So can we estimate biological age not just with epigenetic marks, but with other things? And to get a better estimate of what does it actually mean relative to advanced biological age. Does that mean you're going to die earlier? You're going to develop type 2 diabetes? And it could be a function of what your genetic background is. Advanced biological age may make you more susceptible to certain disease outcomes. Does that answer your question?
Yes, thank you.
Okay, other questions, comments? We must be getting, oh, Fred?
Great talk. I was just curious, like a lot of what you're presenting is from a public framework on how this is going to be funded. I'm just a little bit wondering, like, how's the private or community partners kind of fit into this with funding mechanisms, training? Because a lot of the current, R01s or Ps don't necessarily think about it that way. And I guess then part two question is like the training. We talked a lot about multidisciplinary scientists and, maybe the traditional silos that we have through departments and structures at the med schools or other places isn't going to be, amenable to what you're thinking. So I'm just curious, from your perch, what do you see?
Well, let me start with the first question, okay, which is one that I'm personally very interested in. So let me take the issue of climate change and health, the health effects of climate change. Okay, so we're changing the paradigm of this now. So it's not just using the $40 million that we got from Congress at NIEHS to fund a few R01s. We're now using that in an innovative new way to essentially provide catalytic funding to get greater interest across the entire NIH, to develop potential interest in the health effects of climate change, and then people can be drawing from their base budgets. So I know Richard Hodis at the National Institute on Aging is very interested in some of these health effects associated with climate change. But it shouldn't be just the NIH. So where's the Department of Defense? Where's the EPA? Where are other federal agencies? And furthermore, what about the Wellcome Trust? So Wellcome Trust has a big checkbook in their back pocket. And it turns out that there are 3 areas of scientific focus for the Wellcome Trust, and one of them is studying the health effects of climate change. So what I've been doing is, in fact, not me, but and I failed to mention that Gwen Coleman is an epidemiologist, a very accomplished epidemiologist at NIEHS. I mean, I moved her into a different role at NIEHS. She's in the office of the director. She used to be in charge of the extramural research division, handing out all that money to you and others. But now she's there in the office of the director with me, and her job is to coordinate this effort. So she meets with the folks at the Wellcome Trust. She meets with the folk at the Rockefeller Center, at the Rockefeller Foundation. She meets with the folks with Burroughs Wellcome. And then the question is, how can we develop a global strategy of funding important things that need to get done in studying the health effects of climate change? And let's do this in a coordinated fashion. So everyone doesn't get out and start spending a whole bunch of money quickly because we got to get it done by the end of the fiscal year. But we do this in a way that we understand what we do well at the NIH. What does the Wellcome Trust do well? I mean, the advantage of being in a philanthropy, they got a checkbook in their back pocket. They can write a check at any point. So what can they be doing that it's harder for us to do? So that's what we're trying to do, is bring greater awareness. So Gwen is, in fact, I think she's currently at the University of Pittsburgh at a conference on climate change and health. She just got back from South Africa. So trying to get global awareness, what can we be doing collaboratively together? Because we have different organizations with money, and how can we be doing this in a more concerted way? Victor Zhao at the National Academy of Medicine is also very interested in this. I've encouraged Victor, and he's done this to some extent already, let's have some National Academy meetings on this. Let's bring together the Wellcome Trust. Let's bring together, philanthropists who are interested in the topic area and see if then the National Academy can be working to develop these collaborative frameworks and how we can be working together in synergistic ways rather than everyone just kind of getting out there and just doing what we have to do. Does that answer your question?
Yeah. And I just think that maybe the training mechanism for young scientists, that, we train them through silos, so it's hard for them, or I think it would be hard for them to come out and be able to see this bigger.
Well, that's a really loaded question. We started talking about this, but training scientists, in fact, I went through this very quickly because I didn't have time, but take a look at the climateandhealth.nih.gov, and there's a training component there. And so we as scientists, more senior scientists, need to be working collaboratively together, but we need to be training our young investigators to work in a collaborative mode. And that's very different than the way I was trained. Not that the training here at Case Western Reserve wasn't great, it was just a different time. So it's all about kind of training someone to become a PI who works to bring in postdocs and graduate students doing things in your own lab. Doing things collaboratively is just more complicated. So in some ways it's a culture change. So recognizing that if we want to take on large complex problems, we have to be seeking out people who can do things that we can't do. And then developing, I think we talked about this, developing collaborative strategies. What can you do? I use the example of polycystic kidney disease. I'm a geneticist. I don't need to collaborate with people who do genetics. I'm actually pretty good at that. But finding people who do the pathology of polycystic kidney disease, Kathy Carlin and others that do the cell biology. I mean, when you get people with different capabilities coming together, magic can happen. In fact, in the case of polycystic kidney disease, we started developing hypotheses that no one would have thought of doing on their own. And so we need to, in our culture, in our training culture, Dr. Gerson, here at the medical school, teaching people how to grow up in a collaborative environment. And partly doing, I think, part of what we did when I was here, with Tony was up in biochemistry and I was in molecular biology. The physical boundaries really didn't inhibit us from meeting on a regular basis and talking about things. My learning about their approaches to what they do and his learning about what we do, it provides for a very, very rich environment, but we don't do enough of that. And I think we need to really integrate that into our T32s and the type of training opportunities we provide our students. It looks like you have something else to say. This is an important topic. Could we give Fred the microphone again?
No, I think that was, I think you answered it. I was just thinking about some of the, the T32s and reviewers don't always see it the way you just described. And so,
Well,
that's an obstacle that gets really challenging to overcome. And, I don't know if that is a direct down approach that needs to be or something to think about, or if it's something that's just going to have to be more organic and grow up.
Well, I think it's about communication. So that's when I talk about our strategic plan, I try to emphasize this isn't Rick's strategic plan. It's not something I came up with in my office with a closed door. It's we're reaching out to the global community. What are the things we need to be doing? So then taking that and letting people know what are the strategic directions. And then study sections need to pay attention to this as well. And so in fact, just as a little detail here, so we have funding, we'll probably fund up to, say, the 11th and 12th percentile with total dollars. But in reality, we publish the 7th percentile. So if you get in within the 7th percentile, you're pretty much in, okay? But between the 7th and, say, the 15th percentile, we have a strategic plan. So that gives me, ultimately, the ability to look at the strategic plan and ask the question, is a 12th percentile grant, does it better meet our needs, according to our strategic plan, than the 9th percentile grant? So it gives me the flexibility in this gray zone to selectively fund proposals that are in alignment with the strategic plan. So there's a little bit of flexibility there. So sometimes we will reach out a little further than 15th percentile, but it really has to be in the context of alignment with the strategic plan. So I don't know if that was helpful or not, but it's...
So with that, I want to first of all, thank Rick for his formal presentation, and even more for a very, very rich question and answer dialogue. I think we all learned a lot during that, and I want to help us continue it. We've got a time for networking outside for, I won't say off the record, but conversation.
Okay, yep.
That can always be helpful. And on your last comment, of course, all grants are created equal. Some are more equal than others, but thank you very much for the effort and the openness with your future sense of where
That's good. Great. Well,
country
again,
science
thanks for the
needs to go.
opportunity to be here.
Glad
Appreciate it.
you're alive.